that you are live. Good morning, 11 o'clock. And on the fifth Sunday, it's time for a special Chai and Why, where we are live from a lab in TIFR. With me is Professor Koti, and we are in his lab, which is the protein mechanics lab. Now, I thought proteins were something we ate.
Yes,
You know,
yes,
get
yes,
muscle
yes,
and
yes, yes,
eat
yes,
protein.
yes. We are going to
And
build
mechanics
muscle.
is all, stretching things and
Yes,
bouncing balls and
absolutely.
springs and all that. So you combine all this.
Yes, yes, yes.
Okay. So sounds very exciting. Protein mechanics. So this looks like, as a lot of TFR, a very, very interdisciplinary lab that works at the interface of physics, chemistry, and biology. And I think I'm going to hand it over to Koti. Koti, you can introduce the students who will be taking us through the lab today, and maybe tell us a little bit about what you're doing first.
Yes,
Because
yes.
I don't think people know about protein mechanics. I mean,
Yeah.
I think protein is some stuff you eat in the morning.
Correct. Yes. So
Okay,
first, yeah.
so over to you.
So thank you, Arnab. And first of all, good morning to everyone. I'd like to thank Chai and Wai for choosing our lab for this Up Close with Science session. And I'd like to thank especially Arnab and Ullas for this session. So what we are going to do today is that we are going to give a virtual tour of our lab and then show a little bit about our instrumentation, where we work and what we do, and then we'll be very happy to showcase that lab. So very briefly, our lab works is an interdisciplinary, combines themes of chemistry, biology, and physics. Chemistry and biology, where we combine and work in the area of protein biochemistry, and then we combine with the physics aspects of force mechanics. So that's why we marry protein biochemistry and force mechanics to bring together protein mechanics. So with that very short introduction, I'd like to invite my lab members one at a time. Suman, so research scholars.
One sec, there is some camera issue. Just one sec, we'll have to go back a bit. Because on YouTube, we are seeing something different. So, why is this camera on? This is camera on.
Sorry for the interruption. We are going to resume soon.
Yeah.
Okay, so I'm
sorry for that.
Yeah, so I'm going to just tell very briefly about what is protein mechanics about. So our lab actually combines research themes, chemistry, biology, and physics. We combine chemistry and biology to put it to protein biochemistry and then combine with physics, force mechanics. So we marry protein biochemistry and force mechanics to do protein mechanics. So with that brief overview about introduction, I'd like to welcome the team of our lab. The research scholars, Suman Tiwari, Aditya Srivastava, Kaushik Chalki, and then Ankur Chowdhury, Samrose Lubanchi, and scientific officer Satyanarayana. So this is, we are a bunch of scientists, research scholars, and scientific officers. So we are going to talk about what we do in our lab. So we'll be very happy to showcase our lab and then tell a little bit about our work. So I'll begin with showing a picture. So showing a picture is this. What you see is on the left is a body muscle, and then on the right spider cell. So there is a common element between these two aspects, is that proteins. So proteins are the important biomolecules which present in all living systems, including animals and plants. And so proteins are almost every function in the human biology can be really carried out these functions. So the picture on the left, you see, it's basically an arm showcasing muscle, And so this kind of muscle you can really build by going to gym. It gives the strength, physical strength. It showcases physical strength. On the right, you see spider silk. It's a beautiful network of spider silk, even adorning small water droplets, showcasing that basically the strength of this web. So, what is a common thing between the muscle and spider silk is that both actually consists of protein molecules, and but their composition is completely different. So, muscle is important. For example, let's say when I stand here or walk or run, right? So, it's a very, very important thing. And at the same time, the proteins in spider silk also are actually very, very important. The common thing of muscle and spider silk is that both exhibit enormous physical strength, toughness, rigidity, hardness, elasticity. But let's delve into a little bit more about what is muscle. Okay, why muscle is actually important. So we have in body a lot of muscle. So typically adult human body consists especially 30 to 40% by body weight consists of muscle. And we have different types of muscles. For example, like muscle in your arms and legs, which is important for walking, running, etc. And the muscle in your heart is very, very important, called cardiac muscle, very important for pumping action. And there is muscle in your stomach, which is important for digesting food. For example, let's say without that muscle, you will not be able to digest your food. So the muscle in the stomach, called smooth muscle, will help you churn your food, fragment into smaller fragments so that enzymes can really digest, so that you take your nutrients in the body. And at the same time, so let's delve into spider silk. So spider silk again consists of protein as their material, and then the protein composition in spiders is different from the muscle proteins present in your body. So, for example, like spiders, they spew the spider as a liquid jet, and then this liquid jet solidifies in air, becoming this beautiful spider cobwebs. And such webs actually are completely made up of protein based materials. And interesting thing is that, so because the muscle, human muscle in ours versus spider silk, both consist of protein based materials, and these materials actually provide these natural systems with incredible strength, toughness, mechanical stability, mechanical properties, hardness, and elasticity and tensile strength. Everything you can really think of in terms of physical and mechanical strength. The question is that what makes these natural systems get their incredible mechanical stability, the physical strength? Our proteins are actually the central actors in these systems. So these are the proteins actually which we study in our lab. So what we do in our lab is we think proteins as simple mechanical springs. Okay, just like what you see the spring pictures in your school textbooks, we believe these proteins are the tiniest of tiniest springs present in natural systems, which actually function as force sensors. So these molecules can actually sense forces or exert forces both in muscular systems or provide incredible strength in the spider cell. And so day to day, we make these proteins in the lab using variety of synthetic and biological methods, which my team members are going to introduce to you. At the same time, we actually study these proteins like molecular springs using advanced experimental methods developed in our laboratory. So we are going to showcase some of these methods, which are based on atomic force microscopy based tools, where actually we build these instruments to do the experiments, the kind of experiments which we want to do, where imagine like you can really hold single molecules of proteins. These are the very tiny, tiny objects, and then apply very tiny amount of forces, and then visualize them as springs, the spring balances which you typically use to measure the weight or the force. But we use atomic force based methods in the lab to do these experiments. So with that very short introduction, our lab team members are going to take you through how we actually make proteins, what are the proteins, how we make them, how we characterize them, and also importantly, how actually we use experimental methods developed in the lab to do these sophisticated pulling experiments on individual molecules so that we study their mechanical properties. Okay, with that, I'm going to pass it on to Lubanshi, the member in the team. She's going to talk about how we actually make proteins in the lab. Lubanshi?
Hello everyone, this is Lubhanshi. I'm a fifth year graduate student in Koti's lab, and I'm going to walk you through what are proteins, how do we came to study or investigate today as we do, and what what lead us to this current state of our techniques that we utilize day to day in our lab. So I'm going to show you some slides for the same. So the term protein is actually derived from a Greek word. So the term protein is actually derived from a Greek word, proteos, meaning of primary importance. And the study of proteins actually started 200 years back, where people were actually trying to understand different life forms that existed around them, and they used to see materials.
Going to the slideshow.
Sorry. And they used to study different forms of life or food items that were available to them, such as eggs or milk, or even they used to study muscles, tissues from animals or humans alike from patients. And they used to come across a substance which was present in all the different life forms, and it was reactive to heat and acid treatment. And they came to a conclusion that this is a substance which is present in all different life forms and is somehow regulating the core of the system that is functioning. And then came the famous experiments by Mendel, where he explained inheritance, where he came to the conclusion that the offspring carries characteristics from its parents. But still the clue was not, still we didn't know what is the actual reason that an offspring is similar to their parent, or what is the information that is carried down from generation to generation. And here comes the discovery of DNA. The structure of DNA was discovered by by Rosalind Franklin and Francis and Crick, and it came to be known that this was the missing link between protein and the hereditary information that is carried from parents to offsprings. So DNA was the hereditary material, the genetic code of life, where it carries all the information that is required to build an organism. But how did we used to study proteins back then when we didn't know that DNA makes protein? So this was a very crude way to study proteins in the age of like in 19th century or 1800s, where actually the tissues or blood was used to extract proteins from the crude muscle. And to get purified protein, it used to take very hard work and the actual animal tissue. So it was very limited way to get protein, and it was also very rigorous and also not very feasible because of ethical and technical reasons. That limited the study of proteins for a very long time. And now we come to the modern age where we can express proteins in our lab. So this took about a decade, and a lot of discoveries in a series of events where first DNA ligase was isolated, which is a protein that joins DNA molecules together. And then the first recombinant DNA molecule was synthesized by Paul Berg, which lead to the discovery that we can actually synthesize a protein of interest in an organism where we can get the purified protein not from a tissue but in a lab. And this is how we, in today's lab, we actually express protein. And these all discoveries are of great importance because this is how we actually know. So you all must be knowing that diabetes is a very common issue in today's world. And insulin is the cure for diabetes because is a treatment for diabetes where we need to provide insulin to the patient so that he can so that their blood glucose levels can be regulated. So before the discovery of this recombinant DNA technology, the insulin had to be extracted from animal tissues. But now we can synthesize insulin in the lab, and this leads to better availability, and it even led to reduction in costs by 50%. And this is a clear example of how the curiosity-led research led from bench to bedside. And this was one of the major biochemical innovations in the last century. So with this, I'm going to move to what actually consists of a protein. And protein is actually a structure of amino acids joined together in a fashion to make different chain lengths. So is a polymer of amino acids. So there are 20 amino acids and all joined together in a particular sequence to form a protein. Now, we need to synthesize this protein. So what we do actually, we can synthesize it like one by one, attaching one amino acid to one particular another amino acid, or we can provide the sample, the sequence of DNA, because we know कि DNA से protein बनता है, then we can supply a plasmid DNA to a bacteria which will then grow. हम उसको खाना देंगे, उसको सही टेम्परेचर देंगे जिस पे वो ग्रो करेगा, and then we can get our protein by lysing the cell. Basically सेल को तोड़ के उसके अंदर जो प्रोटीन है उसको हम निकाल लेते हैं. So that is how we do it today in our lab. And so this is the way actually information passes from DNA to protein. So this I'm showing this slide where DNA is actually the information, and to break down that information, the information is converted to mRNA, and which is then carried to the ribosome, which is it's like a factory where you can synthesize proteins. And then the proteins get synthesized from ribosome and in the organism. तो जो हमने बैक्टीरिया को डीएनए दिया, उससे हमारा प्रोटीन बनाया, and वो प्रोटीन हम प्योरीफाई करके यूज करते हैं. Now one of my friends, Kaushik, will walk you through the lab and show how we actually do these things in the lab.
Okay. Hello, I'm Kaushik. So I guess I'm visible. Can anyone? Yeah, am I visible now?
Go ahead.
Yeah, okay. So hi, I'm Kaushik. I'm a fourth year graduate student in this lab. So by the time you get to know that we synthesize protein from a living organism, and this is our lab where we synthesize proteins, we play with bacteria, we culture them, we give them food, and then we grow, and finally we extract protein from themselves. So I will show where from we start and where we end. So first we start from this freezer. You can see the temperature of this freezer is -79. So normal freezer in our household, we use, it goes like 0 degrees Celsius where everything freezes. But this is going even 80 times less. So here we show, I will just open this once and you will see we keep our samples here. So these are the samples we kept. Here we keep our cells, our proteins, our pellets, everything. So we start from here. We take cells, which is bacteria, and then we start feeding them. So now I will show you the place where we exactly do all these things. So this is this place. I will give a brief introduction of every instrument. So first, this instrument, this is a biosafety cabinet. This is called laminar airflow. So in this cabinet, inside this, it's a very sterile environment, and you can see this below thing where this thing from where we are having unidirectional uniform airflow coming out, so that no part, so it prevents any particle going inside. So whenever we keep our hands inside, first we always sterilize with sanitizer. I guess you already have, I mean, you have an idea about sanitizer because of this COVID pandemic, which we had earlier. So this is how we do things inside this. Now we have this incubator. So you can see the temperature 37. So this is the optimum temperature where we grow our bacteria. And also this is incubator, and this is called shaker and incubator. So you can see something is shaking, and also the temperature, a fixed temperature is there. So here the bacteria are dancing, and actually they are growing, they are dividing. Now you can see you came to know that we give food to the bacteria. So this is the food. We call it medium. There are different types of medium for different types of bacteria. And now, so here we grow them, and finally we lyse them to get the protein. So this is the machine where we lyse them. You can see there is a probe going inside the sample, and we start it to break the bacteria. So just I showed them everything. Now I will just explain a brief. So we start. So from this incubator, we take out our sample. It looks like this. It's a plate. So you can see there are some dots, some scratches, and every dot actually denotes one bacterial colony, which means it starts from one bacteria and then grows to a colony. So we pick up one bacteria and put them inside their food, and finally grow them in 37 degrees Celsius. So we grow them in this amount. It's a 1 liter media. So we grow them in 1 liter culture. And it takes, like, for E. coli bacteria, it takes 20 minutes to divide. So we grow them overnight and provide temperature. So it grows with synthesizing our proteins. And finally, from this bacteria, we settle down all the bacteria, and we got the pellet. And then from this pellet, we go to the sonicator where we lyse it. So you can see this is the probe, and this is the sample. So here we put our bacteria with detergent, which will lyse or which will rupture the bacteria, and then we put the probe inside, and then we start it. So you can hear some sound. It means it is lysing the bacteria, and our proteins are coming back. So now, how will we be sure that our proteins get expressed? So we obviously will do some characterization, but there is a protein called GFP, which itself grows. So for that protein, we'll have a very unique process to see whether our bacteria synthesize the proteins or not. So here you see that we have two pellets. One is this, and one is this. So there are two pellets, but one of the bacteria grows GFP. So what I will do, I will put UV, and you will see one cell will glow and one will not glow. So you can see your left one is glowing green, but the right one is not glowing under UV, which means we put the we put the DNA, that's the protein making information, in the left bacteria. So it grows GFP and it glows now. But in the right side, we put another protein which does not glow, so it does not glow. So this is where we get our protein. But think, bacteria also need some proteins to survive. So these proteins will get mixed with our protein of interest. So finally, we need some purification technique where we only get our protein and leave the bacterial protein. So I will just take you to that instrument where we purify our protein. So we keep it in another room. This is called a column purification. You might have heard some chalk experiments you might have done in school. So this is the instrument. This is the column from where the protein goes down. And then we get our purified proteins. So I showed you the -80 temperature from -80 freezer from where we start. And I'm showing you the last instrument where we end. And finally, we get our protein. So this is the journey from a bacteria, from a single cell bacterial colony to our desired protein. That's all from my side. Yeah. So now I will hand over to another of my colleague who will tell you the structure, the stability of the protein.
Kaushik, can we take a question?
Yeah, sure.
Okay, so the question is, can we keep 1 liter of medium on shaker?
Yeah, we can keep 1 liter of medium in the shaker.
Okay, okay. So I think so. Thank you. Thank
Yeah.
you. Thank you, Kaushik and Suman. Go to the slides.
Where? Can you please help? Click. Okay. Where I'm saying, yeah. Hi, I am Samrose. I was born and brought up in Bihar. I am currently a PhD student in this lab. So, as Kaushik Bhaiya beautifully explained and show some glimpse of how we synthesize protein in our lab. So, I will take you through in more details so that you can appreciate what experiment we have chosen to show you. So, as we all know that protein in our food, we say that proteins is very necessary for our growth and things. So proteins are we know proteins, but we don't know what is the actual structure of it. As Lubhanshi Deep mentioned that proteins are made up of amino acids. So let's look how the protein look. So a protein has an amazing structure. So let's begin with how an amino acid looks. It's look like this. It has a carboxylic group and amine group, and there is an alkyl group bonded to the carbon. And as we differ the alkyl group, we say that we have differ the amino acid. So essentially, we have 20 natural amino acid, and from this only 20 amino acid, we have millions of protein which are working in our body every seconds. So let's begin how the proteins all look like. So basically what happens is amino acids sit side by side to each other, forming a long polypeptide chain. I'm saying polypeptide because amino acid joins by a peptide bond. So many amino acid join to form a string made up of beads. It's called we call as a primary structure, and in this primary structure the amino acid interact to form the secondary structure. The secondary structure mainly comprises of alpha helix and beta sheet. You can imagine it as a spring, alpha helix as a spring and beta sheet as a sheet of paper. So when these amino acids form the secondary structure, it further folds to form the tertiary structure. Tertiary structure is mainly the functional form of protein, but sometimes more than one polypeptide chain forms a group, and we call it as a quaternary structure. One example I can give you is hemoglobin and myoglobin. So hemoglobin is a quaternary structure. It is a tetramer,
And tertiary structure is myoglobin. And we all know what hemoglobin and myoglobin is. They are proteins, and hemoglobin is used for oxygen as an oxygen carrier, and for oxygen storage, we have myoglobin in our muscles. So there is a balance of all the force in the protein to make it functional. So, what are the function of proteins? As we know now that proteins are made up of amino acid, proteins are mainly, what are the function of protein? So what happens is, in our body there are millions of protein which are working for us to make our body function. So protein act as an enzyme. It catalyzes the biochemical reaction happens in our body. It acts as a structural support to our body. It acts as a signaling and transmitting molecule from one part of the body to another part. So from proteins are, now I hope that you are convinced that proteins are very important. In spite of these examples, proteins have proteins functions have endless example. So as I told that proteins are folded in a particular manner. So what happens is there is a delicate balance of all force. But once there is some stress condition, the protein unfolds. And what are the stress condition? Under oxidative stress, because we are we do aerobic respiration, so there is oxidative stress happens, or the change in pH condition, or mechanically we unfold the protein or denatures it. This process of formation of folded protein to unfolded protein is called denaturation. So this denaturation process is reversible, either reversible or irreversible. So begin with the reversible thing. So it is a basically enzyme, as I mentioned earlier, that protein act as an enzyme. So difference between enzyme and protein is enzyme is a protein but with active site. So when we add denaturant, denaturant means anything which denatures it unfold it. So basically there is a urea or beta mercaptoethanol or guanidinium hydrochloride which denatures the protein or unfold it. And if it is reversible, when we remove the denaturant, it make the protein fold again and functional again. But sometimes this unfolded protein misfold and misfold to form aggregates, oligomers or fibrils, which are risk factor for our body. Because of that, there are a number of diseases which are harmful forms. Example, Alzheimer's, Parkinson, cystic fibrosis, cataract, the most common example. So there is we have to maintain that. One example I can give you for the mutation of protein. So what does mutation means? Mutations means a change in amino acid. There's a particular sequence of every protein. Every protein has a sequence which they can't change. If there is any change in protein, if there is any change in amino acid, it's called mutation. So as Ruvanshidi mentioned, that from DNA, the codes, RNA codes it, and then protein forms. So if there is a change in DNA base pairs, it will impact the RNA sequence, and which further impact the protein amino acid sequence. As you can see that it is a glutamate, and then finally it mutates to valine. One example I can give you, a very common example, is this sickle cell anemia. Is that when we have normal red blood cell, it is circular in nature. When this mutation takes place from glutamate to valine, it changes its shape, and we call this disease as sickle cell anemia. So this is only one example. There are a number of examples where mutation take place and people are health risk of cancers and very different types of dangerous disease. So let's delve more into the denaturation of protein, because we will show you some demonstration regarding this denaturation. So as I mentioned earlier, that when denaturation takes place, protein lost its original shape and lost its function also. So so to convince you, I will give you some examples regarding the denaturation from our daily life, which we all are seeing, but we don't know that what is happening there. So there are three examples: boiling of egg, which we do, I think, every day. So what happen is there is a time period where we have to stop the boiling process so that we get a boiled egg. So after every minute there is some solidification take place, as you can see from this slide. After 10 minutes, it is completely boiled. What happened there is in the molecular level, what happened is the albumin protein denatures, and every time point when we heat, it will completely denature and boil, and our egg get boiled or takes or solidifies. And this is the denaturation due to heating process. But not all denaturation process by heating is irreversible, because we know that once the egg get boiled, it is irreversible. We can't get back to our original egg. But hair straightening, I think all girls can what I can say, all can relate it, that when we straighten our hair, but once it is previously it is curly, and after hair straightening it will become a silky hair. So this is also a denaturation process. Our hair is made up of a protein called keratin. So when we apply heat on them, the disulfide bond which make our hair curly denatures and break the disulfide bond and make it as a silky hair. But after some time, after an hour or so, we get our curly hair back because the disulfide bonds form. So these are the two contradicting examples which I can say that denaturation can happen irreversibly by the same process. We are doing the denaturation by heating, but for boiling egg it is irreversible, and for the hair straightening it is reversible. And one example I can give you, denaturation by acid. This is very common in our household, that we make curd. So what we do is we squeeze a lemon or a pineapple juice to the milk, and after some time it will convert into curd. So the curdling process is an irreversible process, because the protein in the milk casein denatures because of the acidic condition and forms an irreversible denatures and forms irreversible curdling of milk. So with this, I will pass to Aditya, who will demonstrate the denaturation process which we do in lab. So as some glimpse, because we do denaturation in a very large scale and by varying different condition. But for the time limitation, we are doing something to just making sure that you got the flavor. Thank you.
Hi, so I'm Aditya. I did my bachelor's from SDTB Khalsa College, University of Delhi, and then I joined here for integrated master's and PhD program, and currently I'm in my final year. So, as my colleagues told you that we can synthesize any protein in our lab. We don't need to go into the wild. So for example, even if we want to study hemoglobin of a whale, we don't need to dive into ocean and take out some tissue from a whale and then extract that particular protein. We can synthesize using bacteria. All we just need to do, hijack it, its cell machinery, ask it to synthesize our protein of interest, and then we can rupture its cell wall and get out our protein. As Kaushik.
Is it really as simple as you just said?
Yeah, it takes some time and some technicalities are there, but in a nutshell, it's that simple. So for today's demonstration, we are showing you some GFP protein as suggested by its name, green fluorescent protein. It's fluoresce in the green color. So why we have chosen it? Because it's easier to show. It fluoresce in the visible light, so you can easily see its green color. And why it's green? Because of its structure. This is its structure, if you can see, and there is a chromophore inside. And this structure is called beta barrel. You can think of it as a cage which is protecting this particular chromophore from which this green color is coming. And this cage is kind of protecting it from outside world. So now we are going to denature it. We can denature it by various means, but today I'm going to show you either we can denature it by heat. Currently it has this particular shape, and when we denature it, it will get lose its structure, and hence this color will be gone. So we can do by two way. One, I will heat it. So here we have this heat bath, which has temperature of above 100 degrees Celsius, and I'm just keeping it, and we will look it after a few minutes. You will see its color will be gone. And second way, I'm showing you by, so currently you can see it's glowing. Can you, yeah, it's glowing. Samrose, can you hold this from this side maybe, or this side, whatever, the stones. I'm going to add tip box. Can
Can we have the light for a moment?
Yes.
Need to. So what you just said was two ways you are going to demonstrate.
Yeah.
One is by application of heat, and one way is
I'm just adding some chemical. So this is an acid. So what happens inside our body whenever protein wants to get digested, there is lots of HCl in our body which denatures the protein. So it linearizes it so that other enzymes can easily chop it off. Otherwise, it's very packed, hard enzyme. It is hard for enzyme to work with. So now I am adding some amount of acid, and you can see its fluorescence will be gone. I'm just adding a little amount of acid.
Can we have the light on? Can we turn on the lights?
So I'm just adding some amount of acid, and you will see.
And as you add the acid, will it go in a flash or will it go?
Yeah. So it takes some time to denature, but you can see it's more or less instantaneous. Let me just give it a swirl. So you can see it's gone, totally gone. Okay. Can you turn it on the light? So there is some amount of fluorescence is still there because it's not always 100% efficient. So I'll add some amount more of acid. So yeah. So you can see its green color is gone, and there is very small faint amount of fluorescence is there. And now you can turn it on. So you can see its green color is gone. It's faintly green now. And let's see if it has, so it is still greenish color, but you can see the intensity has reduced. You can see it will show some green color. So
the one right now you are showing is chemically treated.
So this one is chemically treated. This
The
one
one in your
left hand is this is chemically, and this is heat treated. So now heat treated one is very simple to reverse. We will just keep it on ice, and we will look after. Just turn it on the light. We will see after a while, and its green color will be recovered.
So is it like the protein gains its original
structure? So unlike in case of egg, what happens, it denatures and gets tangled. So it forms this solid clump. So that's why it's not able to go back to its original structure. But here, there is no formation of any clump. It doesn't get untangled. These molecules stay isolated, and whenever we cool them down, they attains their original structure.
So this should disappoint somebody who wants to make an omelette of a boiled egg.
Yeah. You can't make omelette of all proteins. So yeah, that's the case. So meanwhile, I wanted to give you some, just to show you. So this was our original protein, and this is now denatured protein. And we will look after some time, it should gain some of its original structure. So in this small tube, if you have heard of Avogadro's number, the famous number, which is 6.023 into 10 to the power 23, it has these many number of zeros. So if you can, I have already calculated, in this small vial, there is about 1 milligram of GFP. Okay. So it has about 30 billion molecules only inside of this molecule, inside this tube. So whatever light you were seeing, we are getting from these many molecules. But so I just wanted to give you an analogy. What if you want to study a class full of students which have these billions and billions of students. Now, if I want to measure the height of these students, what I will get a single averaged out number. Or if I ask you what is the color of these students, you will see these individual colors you are seeing, but whatever measurements we are doing, we will get an average picture where this individual information will be lost. So in our next demonstration, we will going to show you that we are going to denature only single protein molecule at a time. You need to appreciate that out of these many molecules, these billions and billions of molecules, we are able to do denaturation of a single protein molecule. So now, yeah.
For the people who fail to appreciate this, you can perhaps look at the number one more time. And then what he is saying is, out of so many molecules, they are able to treat only a single mole or
Single molecule.
single molecule and denature it. So it must be not really as simple as you say again, right?
Yeah.
Okay.
And for the time being, let's see if it has gained. So it has gained some amount of green color, but I guess it's it will take some time. Maybe I gave it too much of heat.
So how
So
much of heat is really required to make
so
it irreversible?
its melting temperature is around 90 degrees Celsius. So if we treat it for a very long time, then it start to get tangled with the other protein molecules. So that's an caveat. So we have to be very cautious about we don't give too much heat and for a very long time. So we'll see after a while, a few minutes. And for time being, our scientific officer Satyanarayan ji, he will tell you how to do these denaturation on a single protein molecule.
Okay.
Hello everyone. So now you have done denaturation of a lot of molecules. Here we will do denaturation using mechanical force. So this is our home built setup, home built atomic force microscope. You might not be seeing a conventional microscope, but this is atomic force microscope, and I will describe in detail all the parts later. But how the experiment is done and what AFM is capable of, we will see here. So imagine AFM as providing you with a nanoscale hand, and which nanoscale hand is capable of stretching a single protein molecule. So that nanoscale hand is called cantilever. So I will show you a real cantilever, how it looks like. So this is a cantilever box containing 10 cantilever chips. You can see easily one. So this is so small, and the cantilever you will not be able to see with naked eye, means with this camera, because that is even much smaller. So I will show you under a magnifying glass how a cantilever looks like and what are the properties and how we are able to stretch protein molecules of nanometer size. So now you see this is actual cantilever. This one small thing which we already see.
Reflecting surface.
Yeah,
Very
reflecting
shiny.
surface. Yeah.
Okay.
So now I will put here. This is called AFM head, atomic force microscope head, and it contains three things. One is the cantilever. One is the light source, laser light. You can see the red color. And third one is a position sensitive photodetector. So now, because we are having magnifying image, now you can see the real cantilever. So if you see, there are total 5 cantilevers, 4 are triangle shape and 1 is rectangular shape. Yeah. So we can show here, we have taken a printout also. Yeah. So this is actually a cantilever. You see this triangular one we use for pulling the proteins. And
so you said there are total 5 cantilevers on a single tip.
yeah.
What we are seeing is only one of the 5.
Yeah. And for every experiment, we use only one of them, depending on our experimental requirement. So now we will move on to how the dimensions are so different. If you see, there is a length difference between all of them, and what is actually used in pulling the proteins. Because what you are seeing is only the cantilever arm. This is not exactly which pulls the cantilever. Cantilever is attached to a tip at the end of this cantilever. If you see here, so this is what we saw just now. And if you zoom it, this is at the end and this is at the tip. This is only 20 micron, if you see.
So the tip is almost 20
Yeah,
nanometer.
20 nanometer. Yeah, sorry,
So
20
So
nanometer.
it's extremely sharp. And
It's
very fine.
extremely sharp, but still, if you compare it with the protein molecules, because protein molecules are even smaller than this, mostly around 5 to 10 nanometer when they are compactly packed. So now you can imagine you are going with the nanoscale hand, and because your hand is so sharp that it can only pick up one or two molecules. So now you can do your nanoscale experiments. So how that is done is, we put proteins on this gold coated cover slip. If you see, this is very shiny and this is real gold, 99.9%. And we put protein sample in physiologically relevant buffer. And from that, this cantilever goes and pick one molecule at a time. Why we don't choose the broad tip? Because if you choose a very broad tip, it will pick up many molecules at a time. But we want to do experiment on single molecule. That's why this tip is very sharp. Now your protein, you imagine it as a spring. It is stuck between two things. One is your gold coated cover slip. Another is your nanoscale hand cantilever. Now you start to pull it. What will happen? First, spring will resist because it is compactly packed, your protein, and after some time, it will give up. And when a protein give up, means its structural, compact structure will give up, and you will get a linear length. Why? Because it will be amino acids only. So there will be length gain in this process, and that length gain is most important thing, because for every protein, this length gain is different. It depends on how many amino acids are there in one protein and how compactly it was packed earlier.
So right now we are basically talking about all nanometer length scales.
Yeah, it's all
So the
nanometer.
Protein
Yeah,
is of nanometer length scale.
yeah,
The lengthening.
yeah. That is also in nanometer. But like it depends on the protein. If you take most of the proteins like 20, 30 nanometer in this range, you will get the length gain. So now you are getting two parameters here in all the AFM. One is length gain if you are doing mechanical experiments, and another is the force. Because your protein is strong, it will pull the cantilever down initially. So that is called force. And another thing is the length. How these are measured in this instrument, I will show you part by part. So length gain measurement is done using this very small thing. This is called piezoelectric positioner. So in our day-to-day life, we have seen how piezoelectricity play an important role in gas lighter in our home where we are cooking every day. So what you do, you put very high pressure on a piezoelectric material and it generates spark.
So
here
are basically inside this casing.
Yeah, inside this casing.
So
yeah.
What is being shown is the casing, because
Yeah, I will show you inside also. Inside we can't open this one, but we have earlier opened and I'm showing you this one. So this is actual piezo which I show you. This is how it is after opening, and this is inside one. So the parts I will explain, but we had to see that piezoelectricity as a phenomena, it can generate resolution in picometer range. So our AFM piezo is capable of having 50 picometer resolution, which is less than the size of an atom. So how it generates? It has lead zirconium titanate as a crystal, and you apply very high voltage and it will contract or it will elongate depending upon the direction of the voltage. So with this, you are able to apply nanoscale length gain measurements. So how that are measured? First, you apply the voltage, and second thing is you should always be able to measure the real distance traveled, because it's a crystal. it might not travel the what we are trying to say it. So it has a capacitance sensor which can actually sense how much we have traveled. So you see, there are three things you can see. One is here, another is here, and inside also you see this one. So these are called capacitance sensor. For every direction X, Y, Z, we have three capacitance sensor. When sensors are there in piezo, it is called closed loop piezo. So for all AFM, whether it is imaging or mechanical force measurement, we use closed loop piezo. So it always gives us the real length traveled. So now we have calculated length gain. Second thing is how to measure the force. So to measure the force, we use a method called beam bounce method. I will turn on the laser. So you can see red light. It's 658 nanometer red light laser. So if you see here, if you can zoom in, I don't know. Yeah. So it is coming from here and it is reflecting from the top of the cantilever. So you can see both of them. So earlier we have seen the cantilever. So cantilever is at the bottom. Now at the top it is gold coated. If you see here, this one. So it's all gold coated cantilever at the top. So why cantilevers are coated? To make this reflection possible in very precise manner. So we have laser, then it is going to a position sensitive photodiode detector. It is in this part. You can see from here also.
Can you shine the light?
Yeah.
Okay.
So even if I am putting this red light, this is not actual laser light, but even if I'm putting this one, you can see that its value is changing because this is also red light and this position sensitive photodetector is able to detect this one. So what happens is, if this cantilever is like this and beam is bouncing, now if cantilever bends, its position will change in this space. And this position is calculated using a position sensitive photodiode detector. And this detector is able to create a resolution of 20 piconewton in our instrument. So now, since we have two things, one is length measurement we have done, force measurement is done. So now the experimental part is complete. You are able to see that protein is unfolded and what parameters we get. We get one is fingerprint marker of every protein, which is called contour length, change in contour length. Because we know total length of a protein by amino acid calculation and compact structure. Now you get two important parameter. One is force, denaturation force, which is a mechanical force of individual molecule, protein molecule. And second thing is you are able to verify whether what protein you have taken is really what you are unfolding here. So this kind of traces you will get in this instrument here. Yeah. So this is schematic. So you see these are the protein molecules. This is a cantilever and this is a laser source. And now when you pull it, you will see that this is rising, force is rising. At this point, protein gave up and now the force decreased because we are gaining length and it has come almost to zero. And then again, there is another protein molecule attached to that. So it will keep on rising and we will get a sawtooth pattern till at the end, when it is completely linearized like this. If your protein is completely linearized, then at the end it will detach either from the gold coated surface or from the cantilever tip. So this is how we do single molecule for spectroscopy experiment. But this nanoscale hand which we are having, this cantilever and this setup, is not only capable of doing this experiment, we can also poke the surface. Why? To measure the elastic properties of the samples like live cells, tissues, and zebra, even live vertebrate animals like zebrafish can be set up. And how we do that is in another instrument. This is also a atomic force microscope, but this atomic force microscope is slightly different because it is coupled with a conventional microscope. So now people will be able to see that this is an actual microscope. But here you see this part is called atomic force microscope, only this part. And the rest is, this one is a conventional microscope or inverted microscope. Yeah, it has a camera and we have a 10x objective. So what is the benefit of this system is, so here, because we are doing with live systems, so you want to exactly land wherever you want. So in this experiment, you are doing fishing. You go, you pull one protein and your experiment is done. But here, if you want to do measurement on live systems, you want to land exactly where your sample is. So this is done using conventional microscope, and then AFM takes care of other parts.
So the conventional microscope is a guiding system.
Yeah, it's a guiding system, and also it can provide complementary information. So here I will show you another type of cantilever. So there you see that that cantilever tip is very sharp. Now what will happen if you have a very big cell of 50 micron size and you are going with a very sharp tip and you are trying to poke it? So its membrane will rupture. So here we need a nanoscale hand, but now instead of very sharp tip, you have a kind of ball. So I will show you that ball, spherical bead we call it, and yeah. So now you see, earlier we saw only this triangular shape because that cantilever was so small that we couldn't see. But now we have a spherical bead, and diameter of this is around 20 micron. So this is very big comparable to the size of cells. So with this we do indentation experiment and we measure the elastic properties of the sample.
That is almost 1,000 times your.
Yeah. So what data we get here is slightly different. If you see, so this is a glass. If you go on glass and try to poke it, what will happen? Force will rise exponentially and you will get more or less a straight line. So what is happening here? Your cantilever is coming and suddenly it is touching the surface. Now because this is very hard surface, this is going straight. Force is rising exponentially, but you are not able to poke in the surface. But here, this is a real zebrafish experiment. And here, if you see, you go and when you touch it, the zebrafish will go inside.
So right now we're talking about the white line.
Yeah, white line.
Okay.
So everywhere in AFM, we will get two traces. One is trace and another is retrace, approach or retract. So you do everything like this. So this is how you calculate the indentation experiment and you measure Young's modulus of these zebrafish and live systems. How that is done, you see, I already showed you this thing. And this is a real zebrafish we have put under anesthesia. And now because it is not able to move due to anesthesia, but it is a live sample, it's not dead sample. And we are going at the head of this zebrafish. Yeah. At the head of the zebrafish, and we are able to do indentation experiments.
So in this experiment, what exactly were you looking at?
It is at the head of the zebrafish.
The head of the zebrafish.
Yeah, zebrafish is mounted in.
Are you looking at the brain of the zebrafish or?
No, only the head. Yeah, only the head.
Head. Oh,
Yeah,
okay. So
yeah.
this is the head from the outside.
Yeah.
Okay.
It's only outside. AFM is a surface topography technique. So everything is happening at the surface. Whenever AFM is touching something, because your nanoscale hand is able to measure the forces only. So everything is happening at the surface. Yeah.
So suppose somebody wants to look at the brain of a zebrafish using AFM.
Yeah.
Is it possible?
You can see the outer topography.
No,
we can image it.
I can cut it.
Yeah.
Then bring you a.
Anything where AFM cantilever can touch, then we can image it. We can do indentation or any kind of mechanical experiment, pulling or something. But it should be directly accessible. It's not a light-based experiment where light penetrates the fluorescent something inside.
So mechanical contact is required.
Yeah, mechanical contact is required. So the third important experiments which we can do with AFM are imaging. So I will show you a simple animation. So now, by now we have understood how AFM works. So AFM has a cantilever and your shining light and position sensitive detector is able to monitor the position. So now if your sample has a rough surface, what will happen? You will go there, and because of piezo, we will be able to monitor its height gain. So at the end, what you will generate is a topography image of height. Means you will be able to see the roughness. So this
Basically get a XY plus Z.
X, Y, Z, yeah, all. But this is at atomic level. If your tip is sharp, atomically sharp, you can do real atomic scale imaging. But we are doing mostly on live systems. I will show you some images here where we have done using this system. And these images, if you see, this is a mica, freshly cleaved mica. Mica is atomically flat. So here, if you see the scale, this is in picometer range, around 200 picometer. So this is G resolution. So this much resolution you can get with this system. This is another interesting thing which we encounter in our daily life, which is called a DVD, which stores data. How it stores data is there are pits and lands in continuous fashion. If you see, I have cut a portion of DVD and I have done AFM imaging on this. So what happens is laser reads data from a DVD by using constructive interference or destructive interference. So whether it is able to read from here or here. So those kind, but it's a real height profile. So you will be able to see that there is a pit and land of around minus 20 to 20, around 40 nanometer. But these all are in picometer nanometer range. Now you do imaging on a real live big vertebrate system, which is called zebrafish. You see here the scale is 50 micron. And these are individual cells you can see. So this is not like a conventional microscopy experiment. It takes around 50 minutes to acquire one image, and this is done using quantitative imaging. But you get additional information, that is the height of these ridges. You can get in AFM, and this experiment is also done live. So with AFM, we have seen how we can do mechanical stretching, you can do indentation studies, and you can image various samples, including live and thickness measurement of nanofilm, etc. So with this, now I hand over to my colleague Ankur, who will talk about protein biomarkers as a disease recognition method.
So can we just take a few questions? So the question is it correct that if the object is large, higher than the force resolution, hence less sharp the cantilever will be? Is that correct?
No.
Okay, let me repeat the question.
Yeah.
The question is,
So
larger the object,
higher the
larger the object, higher the force resolution. Is that correct to start with?
No, force resolution is an instrument property. Whatever you are taking, that will not hamper with the force resolution. Yeah, sharp tip can improve the force resolution. So yeah, obviously atomically flat, if you take one atom, it will provide more resolution in imaging.
So here basically it's a trade-off.
As
far as the force resolution is concerned. But in this experiment, perhaps force resolution, you have adequate resolution. It's not that you are compromising the.
No. Depending on our system. That's why I said cantilever has to be chosen according to the experiment planned.
So your experiment is not really less accurate because you are using a broader cantilever.
No, that is because we are having a cell which is of 50 micron size, you see. So if you take a spherical bead, then only you will be able to measure the elasticity. Otherwise, you will be poking and rupturing the membrane.
I think that question is answered.
Yeah, thank you.
Thank you, Satyaji. So now, by now, you must have understood we are trying to understand the process of protein folding and the kind of forces that are involved during the process of folding and what are the factors that can affect the process of folding in a protein. So we have many tools, spectroscopic tools, by which we try to gauge the properties of a protein molecule. So now I will try to show you a few daily life applications where we are, using these properties of protein to have different applications. So first, I will. So I hope you can see my slides. Yeah. So go to the slide. Yeah. Okay. So first, we are trying to understand the structure and folding and why it is important and where it is coming in use in daily lives. So let's have a view over a few diseases which are very common in the world today. So if I would like to first introduce why it is important to understand the folding. So if we look at we all must have done paper folding trials or okay.
Slide share is fine.
Okay.
Share the slides. I'll float over it.
Okay. Yeah. So we all must have gone through, this origami trials where we try to fold a paper into a certain kind of shape or a bird or a animal creature. Now, if we see here, if there is any step during which the folding goes wrong, then the paper becomes non-useful, and then it is basically becomes trash. Now, similarly, if during the protein folding process, which we are doing every day here in our lab, if during any step there is a misfolding or misalignment happening, the protein also becomes, garbage, and it is no use for the body if it is happening inside the cell. And what happens? These proteins which are becoming non-active, they come together and form these kind of, aggregates which can be toxic and non-toxic depending on the conditions in the cells. So I will show you a few examples where these are becoming toxic for our body, and these are leading to diseases like conditions. So first example is, oh, sorry.
Click on that.
Yeah. So first example is Alzheimer's disease, where we have a protein monomer, which is called A beta protein. And what happens, this A beta protein, when it misaligns with another A beta molecule, what happens? They start, forming these oligomer-like shapes, and they lead to formation of these fiber-like, aggregates in our body, leading to plaques which are formed inside our body in between the neurons which are there in our brain. Now you see here, these are brown shapes here. These are misfolded proteins because of misalignment, and these are very toxic in nature, which can, hamper the functioning of neurons and, deadening of our brain, which is basically a major cause of, memory loss in elderly patients. So what happens? How we can, track this disease? Basically, we can take out the samples from blood and spinal fluids, and we can track the amount of amyloid protein present in the blood and spinal fluids, and we can track the disease situation depending on the patient's stage of the disease. So basically, this helps, the protein helps us in tracking the disease along the process. Another example is Parkinson's, where alpha-synuclein, which is a very commonly known protein in our society, unfolds, and due to environmental conditions have mutations occurring in the protein sequence, which leads to misfolding of the protein and formation of fibril and then Lewy body structures, which are fluid-like structures which are toxic in nature, and they'll lead to synaptic dysfunctioning of the neurons and eventually leading to memory loss. So these are few examples. Now here in this case also, we can track the concentration or the number of monomers to the oligomers to the Lewy bodies formed in the cells by extracting out blood samples, cerebral fluids, and we can track the disease, at what stage the disease, and if there is a probability of progression of the disease. Similarly, we must have seen recent case of COVID, which was all over we all have suffered through the 2-3 years, and we must have seen this the image of this SARS-CoV virus. and it has spikes coming out of the surface, which are basically the spike proteins. And these proteins are basically the main culprit, which leads the binding of this virus to the membrane of a cell and allows it to transfer the genetic material to our cell and causing infection inside our body. So how here we can use the protein structural determination and comes handy. So when we take out the swab from our nose and try to take out the virus, the structural determination of the protein, which is here, you can see here, it helps the structural determination helps in finding out more targets which can bind to these proteins and make it inactive so that we can cure this disease. So it has been very useful in recent times, and many techniques have been developed which directly target this spike protein and make the coronavirus inactive and helps us cure. And similarly, another, one of the most common cancer is breast cancer now. And in what happens in breast cancer is that suppose we have normal cells which are there in our body. Now these cells have this HERP2 growth factors, which are proteins here. Now these are responsible for division of the cells. And if by mutation we have a lot of these proteins on the cell, then what happened? The division of the cell will become very fast, and which will lead to formation of these cancer-like metastases, which will lead to devastating effects. So we can, what we can do, once we have the structure of the proteins by using different methods, we can target these proteins and make them inactive using antibodies, nanobodies, and different methods, and stop the progression of the cancer by different modes. So these are a few applications which can be, come in handy, and they are becoming very useful in today's world, which basically originate from structural determination and mechanical property determination of protein. So now I would like to transfer to Suman Tiwari, who will also give you a few beautiful examples where this is coming in handy. So
Stop the slideshow.
Yeah, sure.
Am I audible? Okay. So, hello, and this is the last talk. So thus far you have heard a lot of proteins and how we study, how we denature them. And the example that you came across was GFP. Now, we didn't just show because we had the protein. I'm going to talk about how GFP, a protein, can actually help diagnosing the diseases which are caused by further proteins. So I have chosen a title that is colorful yet invisible, because GFP comes from jellyfish and it uses this protein to become invisible in ocean. You might have come across this firefly and mushrooms in Western Ghats, which are fluorescent, but they glow in dark for visibility. And they glow because they have some organic compound, which when oxidized in presence of an enzyme, that fluoresces. And that compound is luciferin. And if you look into the structure of luciferin, it is this. So when luciferin converts to oxyluciferin, which is done by an enzyme, these organisms glow, and they glow clearly to gain visibility. But if you look at jellyfish, which is found in the ocean, it glows to become invisible. So first I'll explain that how these organisms become invisible by glowing. If you look at ocean, so sunlight reaches to very tiny depth of the sea, which is to the twilight zone where most of these jellyfish-like organisms are found. So when you look at the organisms in the twilight zone, they appear blue from the top. But if any bigger organism looks at those organisms from the bottom, they look black, and hence they become easily detectable. So what these organisms do is that they start glowing from the bottom side, where now they look blue from the bottom also and blue from the top also, by which they become invisible. This is an example of a fish which does this. This fish found in the twilight zone glows from the bottom and appears blue from both the sides, from the top and from the bottom.
becoming invisible. So I think I've convinced you how a fluorescent protein helps these organisms become invisible. But why are we really talking about this protein? Why is it really that interesting? Because this fluorescence is coming from a protein. As I said, that the organisms like firefly are fluorescent because of a compound, and you cannot take compound and transfer it anywhere. So when this comes from a protein, there has to be a DNA for it. As Samrose explained, Lubhanshi explained that proteins are all made by passing on the information from DNA. And because of this, for this discovery that this fluorescent compound is not a compound but a protein and it has a DNA, these three people were awarded with Nobel Prize. So, you know, thus far that DNA, it becomes mRNA and that becomes protein. So if you have a DNA for GFP, you can easily attach it to the DNA of any organism and you can make GFP in that organism. Like we showed that we make GFP in bacteria. Here we grow it, here we make this protein in bacteria because we want this protein and we want to study the properties of it. But this can be translated to any other animal, and not only in the green color, because we can play with DNA whenever we want. So scientists have made different color mutants of this protein as well, where this particular amino acid Y66 has been mutated, and now we have a range of fluorescent protein, which is not only green, it is blue, it is cyan, it is yellow, it is red. All the fluorescent proteins are there. And all these fluorescent proteins have traveled from jellyfish to animals, where the study is being done on mice. So I have a video where a mice whose DNA is incorporated with the GFP DNA is glowing in dark. But why are scientists doing it? Do they really just want colored animals, or are they really doing it for some purpose? So this was done to see a particular type of cancer which was spreading in the mice. And so what they did was, as Ankur talked about, that some cancers involve overexpression of some proteins. Now that protein is also being made from the DNA. If we attach our GFP DNA to that cancer causing protein's DNA, we can express the fluorescent proteins and we can really see that what part of the mice the GFP has traveled into, or basically what part of the mice the cancer has spread in. Now you can appreciate it, like there are very costly machines to visualize and to understand the information of any disease diagnosis, be it MRI or be it whatever you have seen in a doctor's clinic. But here you can just see it by flashing a UV torch. So it is not only helping us diagnose, it is helping us diagnose it in a cheaper way. This has been translated to humans as well. Well, this is still under clinical trial. This has not been come in market yet, but few pox lesions, their gene therapy, where a vaccine is nothing but a gene, was also added with the GFP gene, and it was seen under UV light that the drug reached in these lesions which was caused by pox, and we could easily spot whether our drug is reaching at the target or not. So GFP basically has traveled a lot. It has traveled from jellyfish to mice to humans, not only just a traveling part, but with a lot of medical applications. And there is a wide range of exploration that is yet to be done with this fluorescent protein. So with this, I would like to conclude my talk. Thank you. So, yes.
So,
Okay. So
so wonderful. Proteins seem to
yes,
be doing
it's
lot many
one.
things.
Yes, they really work out in our lab along with us, and I hope you got overview about our world of proteins, where we make them work, and also they make us work to build muscle and function in biological cells. I hope you have a wonderful time, and we'll be happy to take any questions. You can always post your questions on YouTube, or you can contact us through our website on TIFR. We are in the Department of Chemical Sciences at TIFR.
Yes.
Thank you, Arnab,
Remember,
once again.
thank you, Koti, and thanks everybody in the lab for doing this. Remember, if you still have questions which are not answered live, you can send an email to outreach, outreach at tifr.res.in, and we will forward them to Koti's lab, and you'll probably, can get the answer. Now, before we end, let me just tell you, in case you're watching this, that the next session, which will be on the first Sunday of September at Prithvi Theatre, 11 o'clock, will be by Professor Vidita Vaidya, who's a biologist, a neurobiologist, who will talk about the work she does on understanding fun things that happen in the brain. So that's going to be the next one up at Prithvi Theatre. Till that time, keep watching our videos if you'd like, and feel free to send in your comments and questions. Thank you.